Hello everyone, myself Lokeshwari from 1st MSc Microbiology, Sekadar College, Tirupattur. Here we are going to see about the bacteria Vibrio parahaemolyticus. The general characteristics of Vibrio parahaemolyticus: Gram's classification, gram-negative bacteria, shape, curved rod-shaped bacilli, spores, it is a non-spore forming bacteria, motility, motile, swimming motility. The size ranges from 0.5 to 1 μm in diameter. It belongs to the family Vibrionaceae. The capsules are occasionally present. The endospores are absent. Mode of respiration, facultative anaerobic. Optimum temperature 30 to 35 degrees Celsius and the pH 6.8 to 10.2. Habitat ubiquitous marine bacterium. It is mainly found in the free-living seawater and marine environment. The organisms are halophilic, salt-loving. It was discovered by Tuncero Burro Fugino in the year 1950. Next, the scientific classification of Vibrio parahaemolyticus. The scientific name Vibrio parahaemolyticus and the domain bacteria. It belongs to the family Vibrionaceae, order Vibrionelles and the phylum Pseudomonadata. Pathogenicity of Vibrio parahaemolyticus. First, disease transmission. Consuming the raw and undercooked fish or shellfish, it easily adheres to the living surfaces like aquatic animals. It is exposed to the open wounds. The incubation period of this bacteria ranges from 4 to 30 hours, usually 12 to 24 hours. The generation period 10 to 11 minutes. Next, virulence factor of Vibrio parahaemolyticus. Structural components are capsules, phosphatase, lipopolysaccharides, cholera-toxin, streptolysin, phospholipase and coagulase. Vibrio parahaemolyticus exhibits two toxins. They are TDH and TRH. TDH refers to thermostable direct hemolysin and TRH refers to TDH related hemolysin. Vibrio parahaemolyticus also exhibits enzymes such as gelatinase, lipase, amylase and DNase. The source of infection is by shellfish and the seawater. Mode of transmission is the ingestion of the contaminated water in the seafoods. Vibrio parahaemolyticus is derived from a Latin word and the meaning vibrio, cure, para, side of, hema, blood and the lyticus, dissolving blood. Pathogenesis of Vibrio parahaemolyticus. Infection usually occurs through the fecal oral route and adheres to the host cell with the help of bacterial adhesion factor which are present at the bacterial cell surface. Thermostable direct hemolysin causes the lysis of human erythrocyte cells by binding on the red blood cell membrane. It forms a pore on the surface of the membrane which leads to the penetration of the cell components in RBCs. They also form a toxin called enzymatic activity for its cytotoxicity formed by the TDH and TDH related hemolysin. TDH is responsible for causing toxicity of the cells by forming a channel where extracellular calcium concentration and chlorine secretion increases within the cell. As the osmotic pressure increases in the cell, its morphology, pathology and self-regulation change, causing the cell to expand and eventually die. Another thermolabile hemolysis TLH gene is associated with stimulating the intestinal infection and also causes lysis of the human erythrocytes. Some of the detection methods of Vibrio parahaemolyticus are molecular techniques, biochemical test, PCR based assays, DNA based methods, LAMP, wrapped PCR. Signs and symptoms. It also exhibits watery diarrhea, vomiting, nausea, abdominal cramps, fever, chills, mucus or blood in the stool and even death when the patient internal system changes with the inflammation. The major clinical disease caused by the Vibrio parahaemolyticus are gastroenteritis, wound infection and finally bacteremia. First, gastroenteritis. It is a bacterial digestive problem which is caused by the bacteria. The major symptoms include nausea, vomiting, fever, diarrhea, abdominal cramping, and pain. In most severe cases, they may become dehydrated and may have an electrolyte imbalance. Bacterial gastroenteritis is sometimes treated with antibiotics. It may also last for 2 days. Next, bacteremia. It is defined as the presence of a viable microorganism in the bloodstream and can be categorized as transient, intermediate, or persistent infection. Wound infection. Wound infection can occur if a bacteria enter and multiple inside the wound. Immediately cleaning and dressing the cuts and other small wounds is the best way to prevent infection. However, people with larger, deeper, or more serious wounds should have a trained healthcare professional to treat the injury. Epidemiology of Vibrio parahaemolyticus. It is not easily communicable from person to person, mainly occurs in the summer season, found in the marine environment. Consumption of the contaminated raw shellfish. Bacterial gastroenteritis is mainly found in Japan and Southeast Asia. Seafood associated gastroenteritis is mainly found in the United States. Laboratory diagnosis of Vibrio parahaemolyticus. First, microscopic examination. In gram staining, pink colored gram-negative bacilli is observed. Motility test. The bacteria is motile in nature. Colony morphology on culture medium. In blood agar, it exhibits beta hemolysis, and in the Mechanchi agar, it shows pink colored non-lactose fermenting colonies. Selective medium, TCBS. Thiosulfate, citrate, bile salt, sucrose agar, it exhibits blue-green color colonies. Biochemical test. Catalase test, positive, shows bubble formation. Oxidase test, positive, shows blue color formation. Urease test, positive, pink color formation. Indole test, positive, pinkish red formation. Methyl red test, positive and shows red color. August-Prosker test, negative and shows pinkish red color. Nitrate reduction test is positive and shows pinkish red color. H2S production is negative and exhibits black color formation. Triple sugar ion test is positive and shows yellow bud and red slant. Prevention and control of Vibrio parahaemolyticus. Maintain personal hygiene. Avoid cross-contamination between raw and cooked shellfish. Don't get exposed to the salt water or the brackish water. Avoid using seawater for cooking purpose, and also avoid consumption of raw or undercooked seafood. Treatment. The disease caused by the Vibrio parahaemolyticus is self-limiting. Oral rehydration is required. Drinking plenty of fluids. Taking bed rest. Antibiotic treatment is not usually recommended, but in sensitive cases, antibiotic, mainly tetracycline, is recommended. Thank you everyone.